So the unit the unit 7, I think, genetics and evolution. So we are going to study like three chapters. So principle of inheritance and molecular basis of inheritance, and then evolution. So the work of the Mendel and the others who followed him gave us an idea of the inheritance of patterns. Okay. However, the nature of those factors which determines the phenotype was not very clear. As these factors represent the genetic basis of inheritance, understanding the structure of genetic material and the structural basis of genotype and phenotype conversion became the focus of attention in biology for the next century. Okay, the entire body of molecular biology was a consequent development with major contributions from Watson, Crick, Nirenberg, Khurana, Kornbergs, the father and son. Kornbergs, these are the fathers and son. There are Benzer, Monod, Brenner. Okay, so all these scientists important. A parallel problem being tackled was the mechanism of evolution. Awareness in the area of molecular genetics, structural biology, and bioinformatics have enriched our understanding of the molecular basis of evolution. So in this structure, the structure and function of DNA and the story and the theory of evolution have been examined and explained. Okay, so these are the Watson and Crick, James Watson and Francis Crick. What they did? Okay, now what they did? Okay, they study of the effect of hard X-rays on bacteriophage multiplication. Okay, so so the effect of hard X-rays on bacteriophage multiplication. Okay, then so he was the Watson who did it, X-ray on bacteriophage multiplication. Then Crick, talking about the Crick, he's solving the DNA structure. Now what he did? Okay, the proposal. Okay, the Crick did the proposal of the complementary double helical configuration. Now Crick now what Crick did? Okay, Crick also a thesis entitled X-ray diffraction, that is polypeptides and proteins. Also in thesis he gives entitled X-ray diffraction, polypeptides and proteins. Okay, now together Crick and Watson, they together now they together they leading in 1953 to the proposal of the double helical structure for DNA and the replication scheme. Crick was made an FRC in 1959. Okay, the honors to now in 1959 FRS given was made to the Crick. Okay, so the honors to the Watson and Crick include John Collins Warren Prize of the Massachusetts General Hospital in 1959, the Lasker Award in 1960, the Research Cooperation Prize in 1962, and the above all the Nobel Prize in 1962. Okay, now the principle of inheritance and variation. Okay, now Mendel's laws of inheritance. So these are the contexts. So we are going to study the Mendel's laws of inheritance of one gene, inheritance of two genes, sex determination, mutation, and genetic disorders. So the offsprings identical to their parents, but they also show the differences, right? Now now uh, now the branch of biology known as the genetics, right? So we study all these questions in the genetics. This is the branch of biology. Now this subject deals with the inheritance as well as the variation of characters from parents to offspring. Now what is inheritance? Inheritance is the process by which the characters are passed on from parent to progeny. It is the basis of heredity. Now variation is the degree by which progeny differs from their parents. Now human knew from as early as 800 to 1000 before century that one of the cause of variation was hidden in sexual reproduction. So variation means sexual reproduction, right? Now now uh, now they did also the artificial selection and domestication from ancestral wild cows. We have well known Indian breeds, the example Sahiwal cows in Punjab. Okay, so now we are going to study like the 7 pairs of contrastive traits in pea plant studied by Mendel. So the characters are seed shape, seed color, flower color, pod shape, pod color, flower position, and stem height. So seed shape, that is round and wrinkle. So dominant trait that is dominant, and recessive trait, that is recessive. Okay, so now in seed shape, that is round and winkle. Seed color, that is yellow and green. Okay, so flower color, it is violet and white. So white is a recessive and violet is the dominant. Then pot shape, that is full and constricted, right? Now pot color, in which it has a green and it has a yellow. So green and yellow, you see. The flower position, that is axial and terminal. So axial is the dominant and terminal is the recessive, right? Then stem height, that is the stem height, that is tall and dwarf. So 7 pairs of contrasting traits in pea plant studied by Mendel. So 7 pairs of contrasting traits, right? So 7 pairs of contrasting traits in pea plant studied by Mendel. So the Mendel's laws of inheritance, as you can see. The George Mendel conducted hybridization experiments on garden peas for 7 years, that is 1856 to 1863. So 1856 to 1863 proposed the laws of inheritance in living organisms. So and this was the first time that statistical analysis and mathematical logics were applied in the biology. Okay, his experiment had a large sampling size, which gave greater credibility to the data that he collected. Okay, also the confirmation of his inferences from the experiments on successive generation of his test plants proved that his results pointed to general rules of inheritance rather than being unsubstantial ideas. Okay, so he investigated that the manifestation has two opposing traits, example tall or dwarf plants, yellow or green seeds. Okay, so Mendel conducted such artificial pollination, cross pollination experiments using several true breeding pea lines. A true breeding line is one that, having undergone continuous cell pollination, shows the stable trait inheritance, trait inheritance and the expression for several generations. Okay, the Mendel selected 40 true breeding pea plant varieties as pairs which were similar except for one character with contrasting traits. So Mendel selected 40 true breeding pea plant varieties as pairs which were similar except for one character with contrasting traits. Now some of the contrasting traits selected were smooth or wrinkled seeds. Okay, yellow or green seeds. So smooth or wrinkled seeds, yellow or green seeds, inflated, that is full, or constricted, that is green. So constricted, these are the recessive trait, and full, that is inflated, these are the dominant trait. And then yellow pods and tall or dwarf plants. Okay, inflated or constricted, yellow pods, right? Or green or yellow pods, right? Or tall or dwarf plants. Okay, now the contrasting traits studied by Mendel in pea. Okay, the characters and contrasting traits. So stem height, that is tall or dwarf. Flower color, that is violet or white. So violet is the dominant, white is the recessive. Flower position, axial is the dominant and terminal is a recessive. Pod shape, that is inflated, that is full, and constricted, that is recessive. So pot color, that is green and yellow. Seed shape, that is round and wrinkle, and seed color, that is yellow and green. Okay, opposite it, that is pot color, that is green and yellow. So inheritance of one gene. Now we are going to study the inheritance of one gene. Okay, so he crossed tall and dwarf pea plants, okay, to study the inheritance of one gene. He get the first hybrid generation. This generation is also called the filial one progeny, or the F1. Now Mendel observed that all the F1 progeny plants were tall, like one of its parents. None were dwarf, right? So he made similar observation for the other pairs of traits. He found that the F1 always resembled either one of the parents, and that the trait of the other parent was not seen in them. So he found that the F1 always resembled either one of the parents. Okay, it can be either tall or it can be the dwarf, but it was tall, right? And that trait of the other parent was not seen in them. Okay, so as you can see in here, so steps in making a cross in pea. We need to be emasculated because the flowers are maybe the bisexual or something like that. So you can see the petal, stigma, anther, and stamen and carpel. So removal of anthers, that is emasculation. You can see emasculation from the female parent, I think. So transfer of pollen, then pollination. Okay, so it removes like the anther from the female parent, right? So the steps in making of cross in P. So this is it all. Now the inheritance, as you can see. So Mendel then self pollinated the tall F1 plants. To his surprise, found that in the F2 filial generation some of the offsprings were dwarf. The character that was not seen in the F1 generation was now expressed, so which we were talking like now it is expressed. So he found that the F1 always resembled either one of the parent, and that the trait of the other parent was not seen in them. I mean, the recessive is not seen, but it's now seen. Okay. The proportion of the plants that were dwarf were 1 by 4th of the F2 plants, while 3 by 4th of the F2 plants were tall. The tall and the dwarf traits were identical to their parental type and did not show any blending. That is, all the offsprings were either tall or dwarf, none were of intermediate height. Right? Now, similar results were obtained with the other traits that he studied. Only one of the parental traits was expressed in the F1 generation, while at the F2 both the traits were expressed in the proportion that is 3:1. Okay, the contrasting traits did not show any blending at either F1 or F2 stage. It means both the characters are resembled to their parents. So this is it now. So this is the diagrammatic representation of monohybrid cross. So it is parental, that is tall, and then there is dwarf. So F1 generation it is tall, they get selfing, and in F2 generation it is tall and dwarf. So this is it. Now, based on the observation, Mendel proposed that something was being stably passed down unchanged from parent to offspring through the gametes over successive generations. He called these things as factors. Now we call them as genes. So he called it as factors. Now we call them as genes. Now genes therefore are the units of inheritance. They contain the information that is required to express a particular trait in an organism. So they contain the information that is required to express a particular trait in an organism. So genes which code for a pair of contrasting traits are known as alleles. So genes which code for a pair of contrasting traits, these are the alleles. So they are slightly different forms of the same gene. So they are slightly different forms of the same genes. That is, if you see like if if you are looking at the character that is high, so it can be capital T and small t. Capital T for tall and capital small t for dwarf. So they are slightly different forms of the same gene. Okay, if you use alphabet, okay, so this is all. Like capital T is used for the tall trait, and for example, in case of the character of height, the capital T is used for the tall T trait and small t for the dwarf, and capital T and small t are the alleles of each other. Okay, hence in plants the pair of alleles for height would be capital T, capital small t, and small t. So Mendel also proposed that in a pure breeding to capital tall or dwarf P variety, the allelic pair of genes for height are identical or homozygous, that is capital T or small t. What he did, that is proposed that in a true breeding tall or dwarf P variety, the allelic pair of genes for height are identical or homozygous, right? Capital T or small t respectively. Now capital T or small t are called the genotype of the plant, while the descriptive terms tall or dwarf are the phenotype, right? What then would be the phenotype of a plant that had a genotype capital T small t? So it is heterozygote. As Mendel found that the phenotype of the F1 heterozygote capital T small t to be exactly like the capital T or capital T parent in appearance, he proposed that in a pair of dissimilar factors, one dominates the other, as in the F1 we know, and hence is called the dominant factor, while the other factor is recessive. So in this case, capital T for tallness is dominant over small t for dwarfness, that is recessive. He observed identical behavior for all the other character traits pairs that he studied. Okay. Now in here, like look for the same, like alleles can be similar as in case of homozygotes, like capital T small t. So alleles can be similar as in case of homozygotes and can be dissimilar as in case of heterozygotes, that is capital T and small t. Since the capital T small t plant is heterozygous for genes controlling one character height, it is called the monohybrid. Since the capital T small t plant is heterozygous for genes controlling the one character, capital T small is heterozygous for genes controlling the one character controlling one character height, it is monohybrid. It is single hybrid, that is capital T and small t. Plant is heterozygous and it's controlling and heterozygous for genes controlling one character. So this is monohybrid, and the cross between capital T and small t is a monohybrid cross. So monohybrid and monohybrid cross. So from the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes by the process of meiosis, the alleles of the parental pair separate or segregate from each other and only one allele is transmitted to a gamete. So we can infer that when the tall and dwarf plant produce gametes by the process of meiosis, the alleles of a parental pair separate. The alleles of the parental pair separate or segregate from each other and only one allele is transmitted to a gamete. This segregation of alleles is a random process, and so there is a 50% of chance of a gamete containing either allele as has been verified by the results of the crossings. So in this way the gametes of the tall capital T plants have the allele capital T, and the gametes of the dwarf small t plants have the allele small t. So during fertilization, the two alleles capital T from the one parent, say through the pollen, and small t from the other parent, then through the egg, are united to produce zygotes that have one capital T allele and one small t allele. In other words, the hybrids have capital T small t. Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. The production of gametes by the parents, the formation of the zygotes, the F1 and the F2 plants can be understood from a diagram called Punnett square. It was developed by a British geneticist, Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. The possible gametes are written on the two sides, usually the top row and left columns. Okay. So all possible combinations are represented in boxes below in the squares, which generates a square output form. Okay. The Punnett square shows the parental tall capital T male and dwarf small t female plants, the gametes produced by them, and the F1 capital T small t progeny. The F1 plants of the genotype capital T small t are self-pollinated. The symbols you can see downward for female, upward for male, female that is eggs, and male that is pollen of the F1 generation respectively. Then the F1 plant of the genotype capital T small t, when self-pollinated, produces gametes of the genotype capital T and small t in equal proportion. Okay. So it is all about the 50%. Okay. Okay. So as a result of random fertilization, the resultant zygotes can be of the genotypes capital T small t, and small t. Now, okay, so it can also be solved by a binomial expression, that is ax plus by whole square, because that has the gametes bearing genes capital T or small t in equal frequency of half. Okay. Now the Mendel self-pollinated the F2 plants and found that dwarf F2 plants continued to generate dwarf plants in F3 and F4 generations. And he also concluded that the genotype of the dwarfs was homozygous, that small t. What do you think he would have got had he self-pollinated a tall F2 plant? What do you think he would have got had he self-pollinated a tall F2 plant? So it can be calculated using a mathematical probability by simply looking at the phenotype of a dominant trait, but it is not possible to know the genotype composition, right? We can say through the external appearance that the plant can be that the plant can be tall or dwarf, right? That the plant can be tall or dwarf, right? So but it is not possible to know the genotypic composition, that is, for example, whether a tall plant from F1 or F2 has capital T or capital T small t composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. This is called the test cross. Now in a typical test cross, an organism, that is pea plants here, showing a dominant phenotype and whose genotype is to be determined, that is the dominant phenotype we are going to test, is crossed with the recessive parent instead of self crossing. The progenies of such cross can easily be analyzed, predict the genotype of the test organism. Okay, so the results of the typical test cross where violet color flower V is dominant over white color flower. So using Punnett square, try to find out the nature of the offspring of a test cross. What ratio did you get? Using the genotype of this cross, can you give a general definition of for a test cross? So this is it. Now the diagrammatic representation of a test cross. So based on the observation on a monohybrid process, Mendel proposed two general rules to consolidate his understanding of inheritance in a monohybrid process. Today these rules are called the principles or laws of inheritance. So principles or laws of inheritance. The first law of dominance, the second law or law of inheritance. Okay, so this is the test cross we are going to study. Like, it is a homozygous recessive and it is homozygous recessive, and this is the genotype to which we need to know, that is dominant phenotype, right? Dominant phenotype, that is genotype that is unknown, whose genotype is to be known. Now this is cross, this can be capital W, small w, heterozygous or either homozygous. Now when it is crossed with the homozygous, so it produces all the flowers are violet. Okay, capital W, small w, capital W, small w. So unknown flower is homozygous dominant. If all the flowers are violet, then it is homozygous dominant. If we take the heterozygous, then half of the flowers are violet and half of the flowers are white, so unknown flower is heterozygous, right? So unknown flowers are heterozygous, right? So the law of dominance, the characters are controlled by discrete units called factors. Factors occur in pairs. In a dissimilar pair of factors, one member of the pair dominates, that is dominant the other. So characters are controlled by discrete units called the factors. Now these factors occur in pairs. In a dissimilar pair of factors, one member of the pair dominates the other, that is recessive. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. Okay, so this is the law of dominance. Now going to the second law, that is the law of segregation. So this law is based on the fact that the alleles do not show any blending and both the characters are recovered as such in the F2 generation. Though one of these is not seen at the F1 stage, we are talking about the recessive trait. Though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all the gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. So now we are going to study some exceptions to this Mendel's law, that is the incomplete dominance. So when the experiments on P were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower, that is snapdragon or Antirrhinum sapien. So snapdragon or Antirrhinum. The snapdragon or Antirrhinum sapien is a good example to understand incomplete dominance. So in a cross between the true breeding red flowered, so the true breeding red flower, that is capital R, and true breeding white flower plants, that is small r, the F1 capital R smaller was pink. Now the F1 was pink, right? So when the F1 was self-pollinated, the F2 resulted in the following ratio, that is capital R, small r. So red, pink or white. So 2:1:2. No, 1:2:1. Okay, as you can see in the diagram, that is P generation, red, and then white. So gamete capital R, small r. So all pink, that is capital R, small r. So when they are when they are self-pollinated, like capital R, small r, you can see capital R, small r, and small r. So these are the pink flower. Okay, they are not like red flower, so they are found to be the pink flower. The heterozygotes are pink flower. So phenotypic ratio are red, pink, white. So 1:2:1. and genotypic ratio is same, capital R, capital R, small r, small. So 1 is to 2 is to 1. As you can see, results of monohybrid crossing in plant snapdragon or anti-renal sapium, where one allele is completely dominant over the other, right? So one allele is completely dominant over the other. Now here the genotypes ratio were exactly as we would accept in any Mendelian monohybrid cross, but the phenotypic ratios had changed from the 3:1, that is dominant is to recessive ratio. What happened was that R was not completely dominant over R, and this made it possible to distinguish capital R smaller as being from capital R small and smaller. So explanation of the concept of dominance. Okay, so in a diploid organism there are two copies of each gene, that is as a pair of alleles. Now these two alleles need not always be identical, as in heterozygous one of them may be different due to some changes that it has undergone, which modifies the information that particular allele contains. So these alleles need not to be similar, right? Now these two alleles need not always be identical, as in heterozygous, as you know. So now there are two copies of this gene, the two allelic forms. Okay, let us assume, as is more common, that the normal allele produces the normal enzyme that is needed for the transformation of substrate S, and theoretically the modified allele could be responsible for production of. Okay, so the normal allele that is produces the normal enzyme, but if the allele is modified, now that could be responsible for the production of the normal less efficient enzyme. It can be normal or less efficient enzyme, and it can be a non-functional enzyme or no enzyme at all. So this is it. So in the first case, the modified allele is equivalent to the unmodified allele, that is normal or less efficient, so it can be normal same as now it will produce the same phenotype trait that is result in the transformation of substrate S. Such equivalent allele pairs are very common, but if the allele produces a non-functional enzyme or no enzyme, the phenotypic may be affected. But if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. But if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotypic trait will be dependent on the functioning of the unmodified allele. The phenotype trait will only be dependent on the functioning of the unmodified allele, that is normal allele. The unmodified, that is functioning allele, which represents the original phenotype, is the dominant allele, and this original one is the dominant allele, right? So this one is the so the original phenotype, it is a dominant allele, and the modified allele is generally the recessive allele, and the modified allele, which is, so this is the recessive allele. Okay, this is the weakest allele. Okay, this is not a strong one, this is a weak one. Okay, so recessive allele. Now hence in an example above, the recessive trait is seen due to a non-functional enzyme or because no enzyme is produced. Hence in the example above, the recessive trait is seen. Why it is seen? Because it is due to the non-functional enzyme, or because no enzyme is produced. So the codominance. So till now we were discussing crosses where the F1 where the F1 resembled either of the two parents, that is dominance, or was in, if it was resembling either of the two parents, that is the dominance. So capital T small t, either of the two parents. Okay, in the first first monohybrid cross, as you know, okay, so uh, so so this is the dominance, either of the parents, right? And if it was in between, that is incomplete dominance. If it was in between, that is the incomplete. As you know the examples, as you know that example 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 as I say, like as you know that example that was in between, that is incomplete dominance, that is the pink. But in case of the codominance, the F1 generation resembles both the parents. If it was the codominance, that is capital. Okay, a good example is different types of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by the gene IM. The plasma membrane of the blood group cells has sugar polymers that protrude from its surface, and the kind of the sugar and and the kind of the sugar is controlled by the gene. Okay, the plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. which kind of sugar it is producing, that is A sugar or B sugar, it is determined by the gene I. Now the gene I has a 3 alleles. It is slightly different forms of the same gene. Okay, so it has the 3 alleles, capital A, IA, IB and I. The alleles IA and IB produce a slightly different form of the sugar. You see the kind of sugar it produces, it determines by the gene I. Now while allele I, it does not produce any sugar. So because humans are diploid A, so each person possesses any two of the three I gene alleles. So IA IB are completely dominant over I. In other words, when IA and I are present, only IA expresses because I does not produce any sugar. So IA and IB and I are present, so IB expresses. But when IA and IB are present together, they both expresses their own types of sugar. So this is the examples of the codominance. So hence red blood cells have a so hence the uh, hence red blood cells have both A and B types of sugars. Okay, since there are 3 different alleles, there are 6 different combinations of these 3 alleles that are possible, and therefore a total 6 different genotypes of the ABO blood types. So how many phenotypes are possible? It has like 4 phenotypes. Okay, so 3 alleles, 6 genotypes and 4 phenotypes. So this is how it does. So table showing the genetic basis of blood groups in human population. So IA, you can see IA, so A, IB, so IB and AB. So I, so I, so small I, it does not express anything, so it is I. So IB or A, so I, you can see IB, it is same as IB. Okay, IB it is same, that is AB. So IB it is IB B. So IB small I, it is B, and I it is O. So do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? So you can see that there are more than two, that is 3 alleles governing the same character. Since in an individual any 2 alleles can be present, multiple alleles can be found only when the population studies are made. So there are more than that is 3 alleles, governing the same character. Since in an individual only 2 alleles can be present, multiple alleles can be found only when the population studies are made. Okay, now occasionally a single gene product may produce more than one effect. Now a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. Now it has 2 alleles, that is capital A, small b. So starch is synthesized effectively by capital B homozygotes, and therefore large starch grains are produced. Okay, in contrast, small b homozygotes have lesser efficiency in starch synthesis and produce smaller starch grains. So after maturation of the seeds, capital B, capital C are round, and the small b seeds are wrinkled. Heterozygotes produce round seeds, and so B seems to be dominant allele, but the starch grains produced are of intermediate size in capital B and small b seeds. So if starch grain size is considered as the phenotype, then from this angle the alleles show incomplete dominance. So this is a second example. Of incomplete dominance. It is the like starch grain size. Okay, starch grain size and uh, antirrhinum sapien or snapdragon. So this is an example of the incomplete dominance. So these are the two examples, right? So therefore dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much as on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine, in case more than one phenotype is influenced by the same gene. So therefore dominance is not an autonomous feature of a gene. Or the product that it has information for. It depends as much as on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine, in case more than one phenotype is influenced by the same gene. Okay, now we are going to study the inheritance of two genes, right? So inheritance of two genes. So Mendel also worked with and cross pea plants that differ in two characters. So as in seen in two cross between a pea plant that has seeds with yellow color and round shape, and one that had seeds of green color and wrinkle shape. So seeds that has green color and wrinkle shape, round yellow color and round shape, right? So Mendel found that the seeds resulting from the crossing of the parents had yellow colored and round shaped seeds. How can you tell which of the characters in the pair's yellow green color and round wrinkle shape was dominant? How can you tell which of the characters in the pair's yellow green color and round wrinkle shape was dominant? Thus yellow color was dominant over green and round shape dominant over wrinkles. So these results were identical to those that he got when he made separate monohybrid crosses between yellow and green seeded plants and between round and wrinkle seeded plants. So how do you know which character is dominant when he crosses the yellow seeded plant to the green seeded plants and between the round and wrinkle seeded plants, same as the monohybrid crosses? Okay, so let us use the genotype like capital Y for the dominant yellow seeded color and small y for the recessive green seed color, capital R for round shaped and small r for wrinkle seed shape. The genotype of the parents can be written as capital R, small y, capital R, capital Y, small r, and capital small y. The cross between the two plants can be written down as in the figure. So showing the genotypes of the parent plants, the gametes capital R, small y, capital R, capital Y, small y unite on fertilization to produce the F1 hybrid, that is capital R, small r, capital Y, small y. So when the Mendel self hybridized the F1 plants, he found that 3 by 4 of the F2 plants had yellow seeds and 1 by 4 had green. It is same as the monohybrid cross. So it is same as the monohybrid cross, right? So it is same as the monohybrid cross. Uh, the yellow and green color segregated in a 3:1 ratio. The yellow and green color segregated in a 3:1 ratio. It is same as that of monohybrid cross. Now round and wrinkle seed shape also segregated in a 3:1 ratio, just like in a monohybrid cross. So this is it now. So results of a dihybrid cross where the two parents differed in two pairs of contrasting traits. That is seed color and seed shape, right? Seed color, that is yellow and green. Seed shape, that is round and wrinkle. So as you know, so parental generation, round yellow. So round, capital R, small y, capital Y, that is round yellow, and wrinkle green, that is small r, capital small y. So you saw there is a gamete formation from ry and ry. Now it forms the hybrid, that is round and yellow, capital Y, capital R, small r, capital Y, small y. So now after the selfing, at the F2 generation, as you see, so what are the gametes? Capital Y, capital R, small y, capital R, capital Y, small r, capital Y, capital R, small y, and small y. So these are the same, as you can see. So the phenotypic ratio can be round yellow, round green, wrinkle yellow, wrinkle green. Just remember like that: round yellow, round green, wrinkle yellow, wrinkle green. 9:3:3:1, as you see, small r, small y. So you have to remember this table by the tricks that you make. Okay, now I will make a separate video on this table. Now law of independent assortment. So in the dihybrid cross, the phenotypes round yellow, round yellow, wrinkle yellow, round green, wrinkle green appeared in the ratio that 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination of series of 3 yellow, 1 green with 3 round and 1 wrinkled. As you can see, this derivative can be written as follows, like 3:1, 3:1, that is 3 3s are 9 and 3 1s are 3, then 3 1s are 3 and 3 1 1s are 1. So this is like round yellow, round green, wrinkle yellow, round yellow, wrinkle yellow, round green, wrinkle green. So based on such So based upon such observation on dihybrid crosses, the crosses between the plants differing in two traits, that is Mendel proposed a second set of generalization that we call Mendel's law of independent assortment. The law states that when two pairs of traits when two pairs of traits are combined in a hybrid. When two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. The segregation of one pair of characters is independent of the other pair of characters. So you can see there is a simply so you can see there is a simply so you can see there is a simply monohybrid cross. So there is a simply monohybrid cross. Okay, now so when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters, right? So Punnett square, you see. So genotypes found in F2, they are expected phenotypes. So 9 is to 3 is to 3 is to 1, 1 is to 2 is to 1, 2 is to 2 is to 1. I don't know, man, what type. So, but there is also a genotype or something like that. So chromosomal theory of inheritance. Now we are going to study the chromosomal theory of inheritance. So the Mendel published his work on inheritance of characters in 1865, but for several days it remained unrecognized till 90s. Firstly, communication was not easy. Okay. Now secondly, his concept of genes or factors in Mendel's was as stable and discrete units that control the expression of the trait and of the pair of alleles which did not blend with each other was not accepted by his contemporaries as in explanation for the apparently continuous, because we see the continuous variation in nature. Third, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that factors, genes were discrete units, he could not provide any physical proof for the existence of factors or say that what they were made of. So in 1993, scientists D. Warrens, Correns and Schwann Schermack independently rediscovered Mendel's research on the inheritance of characters also. So by this time, due to advancement in microscopy that we were taking place, scientists were able to make carefully observe cell division. This led to discovery of structures in the nucleus that appear to double. That is the replication of DNA. So appear to double and divide just before the each cell division. So these were called the chromosomes, right? So by 1902, the chromosomes movement during meiosis had been worked out. So the Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel. So the Sutton and Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and use chromosome movement to explain Mendel's law. Recall that you have studied the behavior of chromosomes during mitosis, equational division. Mitosis is equational division because there is a equal division of chromosomes, and during meiosis there is a reduction division because in meiosis 1 there is a reduction of the chromosome number. Okay, so there is a reduction of the chromosome number, right? So this is the during meiosis, that is reduction division. The important things to remember are that chromosomes as well as the genes occur in pairs. Okay, the two alleles of a gene pair are located on a homologous sites on a homologous chromosomes. The two alleles of a gene pair are located on homologous sites on a homologous chromosomes. So this is it. Now meiosis and germ cell formation in cell with 4 chromosomes. Can you see how chromosomes segregate when germ cells are formed? Can you see how chromosomes segregate when germ cells are formed? So these are the germ cells are formed. You see it is G1, then it is G2. You see it is the duplication of chromosomes or duplication of DNA present on the chromosome, but the copies, you see here it is here it is like N. It's like it's the same. uh, Now in the meiosis, that is the formation of bivalent, then meiosis 1, that is anaphase, then meiosis 2, that is anaphase. So meiosis 1, and when they gets reduced in USA, the reduction division, right? The reduction division between the meiosis 1 and meiosis 2. Now meiosis 2, they form the germ cells. So between G2 and meiosis 1 there is simply a duplication, right? So meiosis and germ cell formation in cell with 4 chromosomes. Meiosis and germ cell formation in a cell with 4 chromosomes. Now, a comparison between the behavior of chromosomes and gene. Okay, they occur in pairs. They segregate at the time of gamete formation, such that only one of each pair is transmitted to a gamete. Okay, segregate at gamete formation and only one of the each pair is transmitted to a gamete. Independent pairs segregate independently of each other. One pair segregates independently of another pair. Independent pairs segregate independently of each other. One pair segregates independently of another pair. Can you tell which of these column A or B represent the chromosome and which represents the gene? How do you decide? Almost they all are the same. Okay. So during anaphase of meiosis 1, the two chromosomes pairs can align at the metaphase plate independently of each other. Okay. Now possibility 1, possibility 2. Independent assortment of chromosome in given in here. Okay, so germ cells are formation. So now we are going to study the Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. I repeat it again. Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Okay, the separation of the pair of chromosomes would lead to the segregation of a pair of factors they carry. Sutton united the knowledge of the chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. So Sutton united the knowledge of the chromosomal segregation with Mendelian principles and is called the chromosomal theory of inheritance. Now, following the synthesis of ideas, experimental verification of chromosomal theory of inheritance by Thomas Hunt Morgan, right? So Thomas Hunt Morgan and his colleagues lead to discovering the basis for the variation that sexual reproduction produces, right? Morgan worked with the tiny fruit flies. So tiny fruit flies, that is Drosophila melanogaster. So the tiny fruit flies, that is Drosophila melanogaster, which were found very suitable for such studies. They could be grown on simple synthetic medium in the laboratory. They can be simple in simple synthetic medium. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. They could complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Also, there was a clear differentiation of the sexes, that is, male and female flies are easily distinguishable. Okay. Also, it has many types of hereditary variation that can be seen with low power microscope, right? So it is a Drosophila melanogaster. It is a male that is a small and female that is a large. Okay. Now linkage and recombination. So Morgan carried out several hybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the hybrid crosses carried out by Mendel in peas. For example, Morgan hybridized yellow-bodied, white-eyed females to brown-bodied, red-eyed males and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio deviated very significantly from 9:3:3:1 ratio expected when the two genes are independent. Now Morgan and his group knew that the genes were located on the X chromosomes, and saw quickly that when the two genes in a hybrid cross were situated on the same chromosome, the proportion of the parental gene combinations were much higher than the non-parental type. Okay, because the genes are linked. Okay, Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage to describe the physical association of genes on a chromosome, and the term the combination to describe the generation of non-parental gene combinations, right? So Morgan and his group also found that even when the genes were grouped on the same chromosome, some genes were very tightly linked that showed very low recombination. They were tightly linked, show low recombination, while others very loosely linked, that is show higher recombination if they are placed distantly on the same chromosomes. Okay, when the genes were grouped on the same chromosomes, right? So even after putting on the same chromosomes, there can be the variations, right? So for example, he found that the genes white and yellow were very tightly linked and showed only 1.3% of the recombination. White and yellow, right? And between white and miniature, so white and yellow and white and miniature, that show the variation of 37.2% recombination. Now his student Alfred Stewart went use the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between the genes and map their position of the chromosome. So today genetic maps are exclusively used as a starting point in sequencing of whole genomes, as was done in the case of the human genome sequencing project described later. So this one is cross A cross B. As you can see, this is yellow white. So we see the recombination between white and yellow, it is like 1.2%. As you can see in here, like in the diagram, it is like 1.3%, right? And in the cross is like white and miniature. As you can see, white and as you can see in white and miniature, you see the like W and M, and you can the Y and W, that is yellow and white, it is white and miniature. So you can see like 37.2% of the recombination. And then there is like, here it's given that the linkage results of two dihybrid crosses conducted by Morgan cross issues crossing between gene Y and small and capital small w. Cross B shows crossing between genes small capital W and small capital W and small m. Here dominant wild type alleles are represented with plus sign in the subscription. Okay. The strength of the linkage between the small I and small w is higher than small w and small m. Okay. So this is why the strength of the linkage small capital Y and small y small y and small w is higher than small w and small m, small w and small m. So the polygenic inheritance we are going to study. So this one is the F1 and this one is the parental, this one F1 and F2. So the polygenic inheritance, mental studies mainly describe those traits that have distinct alternative forms. So alternative forms, so distinct alternative forms such as the flower color which are either purple or white. So flower color which are either purple or white. So purple or white. If you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans we don't just have a tall or short people as two distinct alternatives, but a whole range of possible heights. Such traits are generally controlled by three or more genes. Three or more genes, or are thus known as, are called as polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of the environment. So polygenic inheritance takes the influence of the environment. Okay, the human skin color is another classic example for this. In a polygenic trait, the phenotypic reflects the contribution of each allele. That is, the effect of each allele is additive. The effect of each allele is additive. The effect of each allele is additive. To understand this better, let us assume that three genes capital A, capital B, and capital C control the skin color in human with dominant forms capital A, capital B, and small capital C responsible for dark skin color, and the recessive forms small a, small b, small c, these are for the light skin color. Okay, the genotype with all the dominant alleles capital A, capital B, capital B, capital C, capital C will have the darkest skin color, and that with all the recessive alleles, that is small a, small b, small b, small c will have the lightest skin color. Okay, as expected, the genotype with three dominant alleles and three recessive alleles will have an intermediate skin color. In this manner, the number of each type of alleles in a genotype would determine the darkness or lightness of the skin in an individual. So the each allele is very important in the polygenic inheritance of like the human skin height and human skin color, right? So pleiotropy. So we have so far seen that the effect of a gene on a single phenotype or trait. There are, however, instances where a single gene can exhibit multiple phenotypic expression. A single gene can exhibit multiple phenotypic expressions. Such a gene is called the pleiotropic gene. The underlying mechanism of pleiotropy in most cases is the effect of a gene on metabolic pathways which contribute towards different phenotypes. Okay, so the underlying mechanism of a pleiotropy in most cases is the effect of a gene on metabolic pathways which contribute towards the different phenotypes. Okay, an example of this is the phenylketonuria which occurs in humans. The disease is caused by the mutation in the gene that codes for the enzyme phenylalanine hydroxylase, a single gene mutation. This manifests itself through phenotypic expression characterized by mental retardation, reduction in hair and skin pigmentation. So reduction in skin pigmentation, reduction in hair, so all this is caused by like pleiotropy. Okay, it means a single gene affecting the more characters, right? Now we are going to the sex determination, right? So the sex determination, the mechanism of sex determination has always been. So just coming to the point, the Henking in 1891 could trace a specific nuclear structure all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, where the 50% of does not. Okay, Henking gave a name to the structure, that is X body. Now, but he could not explain its significance. So further investigation, this X body of Henking was found to be a chromosome, and that is why it was given the name X chromosome. It was a chromosome, and that is why it was given the name X chromosome. Okay, so it was also observed that in a large number of insects, the mechanism of sex determination is of the exotype. So large number of insects, it is exotype, that is all eggs bear an additional X chromosome besides the other chromosomes, that is also autosomes. On the other hand, some of the sperms bear the X, whereas some do not. So on the other hand, some of the sperms bear the X chromosomes, whereas some do not. All eggs bear an additional X chromosome besides the other. On the other hand, some of the sperms bear the X chromosome, whereas some do not. If the eggs fertilized by the sperm having an X chromosome becomes females, and those fertilized by sperms that do not have X chromosome becomes males. So do you think the number of chromosomes in the male and female are equal? Due to the involvement of the X chromosome in the determination of sex, so due to the involvement of the so what so due to the involvement of the X chromosome in the determination of sex, it was designated to be the sex chromosome, and the rest of the chromosome were named as autosome. So due to the involvement of the X chromosome in the determination of sex, it was designated to be the sex chromosome. Right, so in the determination of the sex, it was designated to be the sex chromosome. So it designated to be the sex chromosome, and the rest of the chromosomes were named as autosomes. Now grasshopper is an example of exotype of sex determination. Okay, in which the males have only one X chromosome besides the autosomes, whereas females have a pair of X chromosomes. So males have only one X chromosome besides the autosomes, whereas females have a pair of X chromosomes, right? So sex chromosome and autosomes and all that, as you can see. So this is the determination of a six by so this is the determination of sex by chromosomal differences. So A and B both in humans and in Drosophila, the female has a pair of XX chromosomes, that is homogametic, and the male has XY, that is heterogametic composition. In many birds, that is female has a pair of dissimilar chromosomes, that is ZW, and male has two, that is ZZ chromosomes, right? So in a number of large number of insects showing the exotype of sex determination, and the rest of them that is the rest of them and the humans. In a number of other insects and mammals including man, exotype of sex determination is seen where both male and female have the same number of chromosomes. Among the males, an X chromosome is present, but its counterpart is distinctly smaller. Among the males, an X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. Females, however, have a pair of X chromosomes. Both males and females bear same number of autosomes. Hence, the males have the autosomes plus XY. Okay, while the females have autosomes plus XX. So, in human beings and in Drosophila, the males have the one X and one Y chromosome, whereas the females have a pair of X chromosomes besides autosomes. Okay. So now, in the above description, you have studied about two types of sex determinant mechanisms, that is exotype and XY type. So XY and XY type. Okay. So both in two cases, males produce two different types of gametes, that is either with or without the X chromosome. So either with or without the X chromosome. It means either with or without the X chromosome. That is either with or without the X chromosome. That is either with or without the X chromosome. Right. If it is XX, that is the male, female, and if it is XO, it means male. If with X or without the XO, type of sex determination, right? It is the male heterogamety. No, it is not. Okay, so this is male heterogamety, and some gametes with X chromosome and some with Y chromosome. Such type of sex determination mechanism is designed to be an example of male heterogamety. Okay, in some other organisms, example birds, a different mechanism of sex determination is observed. So in this case, So in this case the total number of chromosomes is same as in both males and females, okay, but two different types of gametes in terms of the sex chromosomes are produced by the females, that is female heterogamety. So in order to have a distinction with the mechanism of sex determination described above, the two different sex chromosomes of a female bird has been designated to be the ZW chromosomes. Like in these organisms, the females have one Z and W, whereas the males have a pair of Z chromosomes besides the autosomes. Okay, now the sex determination in humans. As you know, 50%, 50%, it is determined the X1, Y. And also even that, or it is also evident that in each pregnancy there is always 50% of probability of either a male or a female child, and but the humans are blamed for this. Okay, and there is an equal probability of fertilizing of the ovum with the sperm carrying either X or Y. Okay, so okay, thus it is evident that it is the genetic makeup of the sperm that determines the sex of the child. Okay, now the sex determination in a honeybee. So you see the parents, the female is diploid, and when it meiosis it is 60-60, and 60 to 60 undergoes a parthenogenesis, that is mitosis, that is male, 60 mitosis 60, and when the fertilization occurs, that is 32. So this is the haplodiplonty life cycle in the honeybee. The sex determination in honeybee is based on the number of sets of chromosome an individual possesses. And offspring formed from the union of a sperm and egg develops as a female, that is queen or worker. So queen and worker, these are the females that can be diploid, and an unfertilized egg develops as a male, that is drone. So male is a drone, queen or worker, it is a female. Okay, by means of parthenogenesis. Parthenogenesis, it is a formation of a it is a formation of a male. So this means that the males have a half the number of chromosomes that of a female. The females are diploid having 32 chromosomes, and the males are haploid, right, having 60 chromosomes. Now this is called the haplodiplontic sex determination system. And as the males produce sperms by mitosis, okay, they do now this is called the haplodiplontic sex determination system, and has special characteristic features such as the males produce sperms by mitosis. They do not have father and thus cannot have sons. They do not have the fathers and thus cannot have the sons, but can have the grandfather and can have the grandsons. So how is the sex determination mechanism different in the birds, and is the sperm or the egg responsible for the sex of the chicks? Okay, yeah, egg is responsible for the sex of the chicks. Now mutation. So what is a mutation? So mutation is a phenomenon which results in the alteration of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. So in addition to recombination, mutation is another phenomenon that leads to variation in DNA. So recombination is also similar to the variation. Okay, now DNA helix runs continuously from one end to the another in each chromatid and in a highly supercoloid form. Therefore loss or deletion, gain, that is insertion or duplication of a segment of DNA results in the alteration in chromosomes. So, since genes are known to be located on the chromosomes, so alteration in chromosomes results in abnormalities or aberration. Okay, since it is a loss or gain of the segment of DNA. Now, since genes that is the part of the DNA. Okay, now these genes are known to be located on chromosomes. See, if there is difference in the alteration in chromosomes results in the alteration in chromosome results in abnormalities or aberrations. Chromosomal aberrations are commonly observed in cancer cells. Okay. Now, mutation also arises due to the change in a single base pair of DNA. This is known as point mutation. Classical example is the sickle cell anemia. So, mutation in sickle cell anemia. So, deletion and insertion of a base pairs of DNA causes frame shift mutation. So, deletion either is to delete or either is to insert. Okay, so these are known as the frame shift mutations. The mechanism of mutation is beyond the scope of this discussion. Okay, but there are many physical and chemical factors that can induce mutations. So these are called the mutagens. So UV radiations can cause mutations in organisms, so it is a mutation. Okay, now as you can see, the male, it is a square, it is a square, then female, that is it is a circle, then sex is unspecified, that is it's called of like triangle, this kind of shape. Then affected individuals, they both are darkened. Mating, you can see there is a line between, and if there are two rods, the mating between the relatives, that is consanguineous mating. Then parents above and children below in order of birth, left to right. So older is present at the left side and younger is present at the right side. The parents with male child affected with disease, as you can see, and 5 unaffected offspring. If we write it inside this kind of square. a different kind of square. So symbols used in human pedigree analysis. So what is pedigree analysis? It is nothing but just a like. So it is just what is the pedigree analysis. So what is a pedigree analysis? What is a pedigree analysis? What is a pedigree analysis? Okay. So since it is evident that control process can be performed in pea plant or other organisms are not possible in case of human beings, study of the family history. Now pedigree analysis, it is the study of the family history about inheritance, that is the characters which are passing from parents to the progeny. So it is the study of the family history about the inheritance of a particular trait that provides an alternative. Such an analysis of traits in a several generations of a family is called the pedigree analysis. So in the pedigree analysis, the inheritance, so in the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generation. Okay, so in human genetics, pedigree study provides a strong tool which is utilized to be traced the inheritance of a specific trait, abnormality or disease. Okay, so in human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality or disease. Some of the important standard symbols used in the pedigree analysis, as I already told you, each and every feature in any organism is controlled by one or the other gene located on the DNA present in the chromosome. So in any organism is controlled by one or the other gene located on the DNA present in the chromosome. So DNA is the carrier of genetic information, right? It is then transmitted from one generation to the another without any change or alteration. So however, changes or alteration do takes place occasionally. Such alteration or change in genetic material is referred to as mutation. A number of disorders in human beings have been found to be associated with the inheritance of change or altered genes or chromosomes. So we are going to study the Mendelian disorders. So broadly, genetic disorders may be grouped into two categories, that is Mendelian disorders or chromosomal disorders. So Mendelian disorders are mainly determined by alteration or mutation in a single gene. Disorders are transmitted to the offspring on the same lines as we have studied in the principle of inheritance. The pattern of inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. So most common and prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia. So it is important to mention here that such Mendelian disorders may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Okay. Similarly, the trait may also be linked to the sex chromosome, as in case of hemophilia. So hemophilia sex link, okay, it is evident that this X-linked recessive trait shows transmission from carrier female to male progeny. So X-linked recessive trait shows transmission from carrier female to the progeny. Okay. Now then there is a discuss with your teacher and design pedigrees for characters linked to both autosomes and sex chromosomes. So representative pedigree analysis of a autosomal dominant trait, for example the myotonic dystrophy, and autosomal recessive trait, for example sickle cell anemia. So you can see this is the trait of the pedigree analysis of the sickle cell anemia. And then there is like autosomal dominant trait, that is for example the myotonic dystrophy. As you can see, the female is affected, okay, the male is affected now, and their progeny. भाई बंद करा भैया, दिमाग खराब करवा छोड़. And female is affected. Representative, okay, and the color blindness. Okay, so color blindness, it is a sex linked recessive disorder due to effect in either red or green cone of eye, resulting in the failure to discriminate between the red and green color. So it is hard to identify the red and green color. So this is the color blindness. It is a sex linked recessive disorder. Okay, now this defect is due to mutation in certain genes. As I know, Mendelian disorder, it is caused by the mutation or alteration, okay, or deletion of a gene. Okay, so this defect is due to the mutation in certain genes present in the X chromosome. Okay, in certain genes. Now it occurs in about 8% of the males and 0.4% in the females. Now why it is more in the male? Because male has only, so this is because the genes that lead to the red green color blindness are on the X chromosome. Because This is because the genes that lead to the red green color blindness are on the X chromosome. Okay, now males have only one X chromosome, so they have the more probability, but female it need to be have the two X chromosomes, so it is the low probability. So it is this, so this is why it is 0.4%. Okay, the son of a woman who carries the gene has a 50% chance of being colorblind. So the son of a woman who carries a gene has a 50% chance of being colorblind. So son of a woman, if woman is carrier, it is like, if woman is carrier, the son has the chances like 50% chances of being colorblind. The mother is not herself colorblind because the gene is recessive. Okay, the mother is not herself colorblind because the gene is recessive. The mother is not herself colorblind because the gene is recessive. Okay, the mother is not herself colorblind because the gene is recessive. We are talking about the heterozygous condition. Okay, that means that its effect is suppressed by her matching dominant normal gene. Okay, and carrier, we are talking about the heterozygous, right? A daughter will not normally be colorblind unless her mother is a carrier and her father is a colorblind. A daughter will not normally be colorblind unless her mother is a carrier, that is 50% carrying the gene for colorblind, and her father is colorblind, that is X, C and Y. That means, okay, so hemophilia, this is X-linked recessive disorder. We already have studied the colorblind, that is sex-linked recessive disorder. Now sex-linked recessive, another disease, that is hemophilia, which shows its transmission from unaffected carrier female to some of the male progeny, has been widely studied. Now in this disease, a single protein that is a part of the cascade of proteins involved in the clotting of blood is affected. Due to this, in an affected individual, a simple cut will result in non-stop bleeding. Okay, the heterozygous female that is carrier for a hemophilia may transmit the disease to sons. Okay, the possibility of a female becoming a hemophilic is extremely rare because mother of such a female has to be at least carrier, and the pedigree and father should be hemophilic. It is same as the colorblind, which is impossible, unavailable in the later stage of life. So this is why the female probability is very, this is why the female probability is very rare. Okay, now the family pedigree of Queen Victoria shows the number of hemophilic descendants as she was a carrier of the disease. Okay, now we are talking about the sickle cell anemia. So this is an autosome link recessive trait that can be transmitted from parents to the offspring when both the parents when both the partners are carrier for the gene or heterozygous. Okay, sickle cell anemia, this is an autosome link recessive trait that can be transmitted from parents to the offspring when both the parents are carrier for the gene or heterozygous. The disease is controlled by a single pair of alleles, that is HBA, HBS, HBA, HBS. So HBA and HBS. So out of the three possible genotypes, only homozygous individuals for HBS show the disease phenotype right, show the disease phenotype. Heterozygous HBA HBS individuals apparently unaffected, but they are carrier of the disease, as there is 50% probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell anemia. Okay, so there is 50% probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. Okay, the defect is caused by the substitution of glutamic acid by valine, that at the sixth position on the beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein results due to the single base substitution at the sixth codon of the beta globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from the biconcave disc to elongated sickle cell like structure. So this is it, like you can see, like, so micrograph of the red blood cells and the amino acid compositions of the relevant portion of beta chain of hemoglobin. So first is the form a normal individual, and second is form an individual with sickle cell anemia. So second is an individual form an individual with sickle cell anemia. So you can see the normal HBA, A gene, GAG, CTC, then mRNA is a GAG. So GAG, that is glutamic acid, it is normal HBA peptide. Now this HBS, it is the infected peptide, or it is like disease peptide. Now you can see sickle cell HBS gene, that is GTG.
GTG, then CAC, then GUG. So GAG is being replaced by the GUG, that is glutamic acid 2 align. So glutamic acid 2 align, right? So this is like, so from an individual with the sickle cell anemia. So this is like, you can see GAG is replaced by the GUG, glutamic acid by valine, at the sixth position of the beta globin chains of the hemoglobin molecule. Now phenylketonuria. So phenylketonuria, this inborn error of metabolism is also inherited as the autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid. So phenylalanine, this group is converted into the phenylpyruvic acid, which is an acid, and other derivatives. The accumulation of this in brain results in mental retardation. So these are excreted through the urine because of its poor absorption by kidney. These are also excreted through urine because of its poor absorption by the kidney. So this is phenylketonuria. It is an inborn error of metabolism. Okay, this is an autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. Okay, so phenylalanine dehydroxylase. So phenylalanine hydroxylase, this is an enzyme. So we are talking about the thalassemia. This is also an autosomal linked recessive disorder. Okay, autosomal linked recessive blood disease transmitted from parents to the offspring when both the parents are unaffected carrier for the gene or heterozygous. Okay, this defect could be due to the either due to due to this defect could be due to either mutation or deletion, which ultimately results in reduced rate of synthesis of one of the globin chains, that is alpha or beta chains that make up the hemoglobin. Now this causes the formation of abnormal hemoglobin. Molecule, resulting into anemia, which is a characteristic of the disease. Now thalassemia can be classified according to which chain of the hemoglobin molecule is affected. In alpha thalassemia, production of alpha globin chain is affected, while in beta thalassemia, production of beta globin chain is affected. So alpha thalassemia is controlled by two closely linked genes, HBA1, HBA2, on chromosome 60 of each parent. Okay, so alpha thalassemia is controlled by two closely linked genes, HBA1, HBA2, on chromosome 60. So on chromosome 60, right, of each parent, and it is observed due to mutation or deletion of one or more of the four genes. Okay, so two closely related genes, one or more of the four genes, right? The more genes affected, the less alpha globin molecules produced. So while beta thalassemia is controlled by a single gene, HBB, on chromosome. So it is controlled by single gene, HBB, on chromosome 11 of each parent, and occurs due to mutation of one or both the genes. Now thalassemia differs from sickle cell anemia in that the former is quantitative problem. So thalassemia is a quantitative problem. It is like the less alpha chain is produced. So it is quantitative problem of synthesizing to few globin molecules, while the latter is a qualitative problem of synthesizing an incorrectly functioning globin. Now chromosomal disorders. So the chromosomal disorders, on the other hand, are caused due to the absence or excess or abnormal arrangement of one or more chromosomes. So it is caused due to the absence or excess or abnormal arrangement of one or more chromosomes. Now, failure of segregation of chromatids during cell division cycle results in the gain or loss of chromosomes, that is aneuploidy. So failure of segregation of chromatids during cell division cycle results in the gain or loss of the chromosomes, that is aneuploidy. For example, Down syndrome results in the gain of extra copy of chromosome 21. Similarly, Turner syndrome results due to loss of an X chromosome in human females. Failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomenon is known as the polyploidy. This condition is often seen in plants. Failure of cytokinesis after telophase stage of cell division results in an increase. Failure of cytokinesis, it means the failure of cell division. failure of the cell division once there is a karyokinesis. So failure of cytokinesis after telophase of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomenon is known as polyploidy. So this condition is seen in plants. The total number of chromosomes in a normal human cell is 46. Out of these, 22 pairs are autosomes and one pair of chromosomes are sex chromosomes. Sometimes, though rarely, either an additional copy of a chromosome may be included in an individual, or an individual may lack one of any one pair of chromosomes. Okay, so these situations are known as a trisomy or monosomy of a chromosome. Trisomy if it is added, or monosomy if it one lacks. So respectively, such situation leads to very serious consequence in individuals. Now Down syndrome, Turner syndrome and Klinefelter syndrome are the common examples of the chromosomal disorders, as you know. So the first one is the Down syndrome. So the cause of this genetic disorder is the presence of an additional copy of the chromosome number, that is 21. So trisomy of 21. This disorder was first described by the Langdon Down, that is 1866. The affected individual is short statured with small round head. The affected individual is small statured. The affected individual is small statured with with a small round head, furrowed tongue and partially open mouth. So furrowed tongue and partially open mouth. Palm is broad with characteristic palm crease. So the palm is broad with characteristic palm crease, and physical, psychomotor and mental development is retarded. So this is the characteristic of Down syndrome. It's too much. Then Klinefelter syndrome. So Klinefelter syndrome, this genetic disorder is also caused due to the presence of an extra copy of X chromosome, resulting into a karyotype of 47, that is XXY. Such an individual has overall masculine development. However, the feminine development, that is development of breast, that is gynecomastia, is also expressed. Such individuals are sterile, right? So it has an overall masculine development. However, the feminine development, that is development of breast, that is gynecomastia, is also expressed. Now the Turner syndrome, such a disorder is caused due to the absence of one of the X chromosome, that is 45 with XO. Such females are sterile, as ovaries are rudimentary besides other features, including lack of the secondary sexual character. So this is the Turner syndrome. Okay, it has a total like 45 with XO type of. It has a total like exotype. Such a disorder is caused due to the absence of one of the X chromosomes, that is 45 with exo type of condition. Okay, with exo type of condition, right? So 45 with exo, and here it is 47, that is XXY. So this is it. So this is all, and here is the like Down syndrome, a representative figure showing an individual inflicted with Down syndrome and the corresponding chromosomes of an individual. You can see the flat back of head, many loops on fingertips like the palm crease, and the broad flat face, and then big and wrinkled tongue. The tongue is big and it is wrinkled. Now the congenital heart disease. Okay, and this is like the 21st chromosome. It has a like trisomy. So this is it, like this is the Down syndrome and Klinefelter syndrome, and this is it. And then there is like the tall statured with feminine character, and short statured and underdeveloped feminine character. It is short statured and underdeveloped feminine character, that is it lacks the secondary sexual character, and tall stature with feminine characters. So diagrammatic representation of genetic disorder due to sex chromosome composition in humans, the first represents the Klinefelter syndrome, and second represents the Turner syndrome, right? So this is it, like okay. So this is it, like and you can see, so these are the chromosomal disorders, right? And so it is all this so it is all the summary and all that. So it is all the summary and all that. So this is all, your chapter is done in here.